disturb you a little bit. I wanted us to just quickly discuss the male reproductive system since I don't have a recorded lecture for this one. I noticed that the attendance is poor, so I'm recording this meeting so that I can share with our colleagues that are not with us. Okay, I hope this won't take more than an hour. Anyway, these are our objectives. So the overview is that so basically in our discussion we will be looking at the reproductive system, which is mainly made up of gonads. So in the males, the gonads are referred to as ova, while in the females, the gametes which are produced by the gonads are referred to as ova, whilst in the males is referred to as sperms. Apart from the gonads, the reproductive system is made up of other components which are regarded as accessory organs, and these are responsible for transporting, nourishing, and storing these gametes. Okay, so we shall focus on the male. So in the male, the gonads are the testes, and their role is to produce sperms, but also hormones. So the other accessory organs, like we said, that are important for transportation of these gametes in the male will include the epididymis, the ductus deferens, the ejaculatory ducts, and the urethra. There are also other accessory organs that are responsible for producing fluids, and these are referred to as accessory glands, namely the seminal vesicles, the prostate gland, and the bulbourethral glands, as we shall discuss in detail later. So apart from the organs and parts of the reproductive system that we have discussed, it is also made up of externally visible structures, which is referred to as external genitalia. So in the male, this includes the scrotum, the penis, the scrotum and the penis. The scrotum is what encloses the testis. The penis is basically the erectile organ that surrounds the distal part of the urethra. This is just a drawing of the male reproductive system that we are discussing. As you can see from the external genitalia, this is something that we are all familiar with. This is obviously the penis and parts of the penis. We can see that this is the scrotum, which is containing the testis, and we can see that from the testis we have the epididymis, and we have a ductus deferens, which is a tubular structure which is moving from the epididymis all the way joining the urethra, even though we can't easily see how it joins the urethra, but I think we can actually. So we can see that it courses behind the urinary bladder. We can see that it then at the point that it meets the duct from the seminal vesicles, it's no longer called the ductus deferens. It is now referred to as the ejaculatory duct, as it passes through the prostate until it finally joins the urethra. And that's of course the ejaculatory duct from the other side of the ductus deferens. Okay, we can zero in on the testis. So the testis, like we've already said, it's the gonads of the male reproductive system or the primary sex organ of the male reproductive system. It is enclosed or hangs within the scrotum. The scrotum is basically a fleshy pouch suspended below the perineum and anterior to the anus. The scrotum, in most people, if not all, is subdivided into chambers, in two chambers or scrotal cavities, and each of these cavities will contain the testis. The testis is described as is described to have a shape of flattened egg or oval shape. Most people say it like that. All right. So we've already said that the testis is housed in the scrotum. The scrotum is basically made up of thin layers of skin and loose connective tissue. And underneath these thin layers of skin is a layer of smooth muscle. This layer of smooth muscle is referred to as the cremaster muscle. The importance of this cremaster muscle is that, first of all, you may want, just to ponder and think. So apparently, why do you think that, or do you know where the testes come from embryologically? Do you have an idea? Are we together, guys, or am I speaking alone?
Following those,
Do you know where the testes come from?
as in the cells or
Or which
are
part of the
they
body? For instance, you know that the kidneys come from the pelvis, and you know that the thyroid comes from the posterior two-thirds of the tongue. So where do the testes come from? Okay, I'm surprised you don't know. So the testes come from the abdomen. So it travels all the way from the abdomen, into, the scrotum. Now, the interesting thing is, why should the testis go to all this trouble of moving from the abdomen into the scrotum? Can you answer that? Madam Tukwiza, are you with us?
uh, what's the question though? Because
Uh,
initially I answered that you came from the abdomen. I don't know if you got me.
no, 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 I didn't hear you. I actually thought I gave the answer, but anyway, it's good if you did. Okay, so a follow-up question. So why, okay, so if they come from the abdomen, why should the body be subjected to, because the process of moving the testes from the abdomen is quite complex. And in fact, sometimes things go wrong. And when they go wrong, that's why we have all those inguinal hernias, because the passage that the testis took, to travel from the abdomen to the scrotum, um, does not close. And if it doesn't close, the loops of bowel can go in there and, become, stuck. And then we have all that intestinal obstruction as a result of, the hernia. So anyway, so what is the point? Why should they move from the abdomen to the scrotum?
I'm not really certain, but I'm thinking since they play a role in the production of time, maybe the temperature.
Sorry, I didn't hear.
Regulate the temperature since they
So it's
to
like your
regulate the temperature
yeah, that's better.
for sperm production.
Yes, apparently, the testes are very sensitive to temperature. In fact, I think when I was in grade 9, I remember quizzing some of my friends, and I think we learned that the the ideal temperature for sperm production, should be lower than that of body temperature by about, I think, 2 degrees also, you know. And unfortunately, if the testes are in the abdomen, not only are you going to have problems producing your spermatozoa, but also these testes could, as a matter of fact, become cancerous, you know. So anyway, so I was talking about the clemaster muscle that you're going to find in the skin of the scrotum. So the role that this clemaster muscle serves is that, if the environmental temperature is high, now as a way of, helping to cool this your testes, what's going to happen is that it's going to relax. And when it relaxes, it's going to move your testis away from the body, helping it, to maintain its optimal temperature for sperm production. And when the environmental temperature, apparently is low, what this clemaster muscle is going to do is that it's going to contract. Does somebody have a question? And when it contracts, yes, you're welcome to speak. If you have a question, please. I don't see any hand. Okay, looks like there's no question.
Join the class. They're trying to join the class.
Okay, shall I proceed? Yeah, so anyway, when the temperature drops, the opposite happens. It contracts and pulls, the testis closer to the body, helping it to maintain optimal temperature for spermatogenesis.
Doc, sorry. There are people trying to join the class. They're trying to join.
Okay, check now. There's no one. Seriously, there's no one. Okay, there's no one. All right, I'm proceeding. So, what was I saying? So I was talking about the testis, but I want to just quickly now give a little bit more detail. So the testis itself is covered by a fibrous capsule. This fibrous capsule is referred to as the tunica albuginea. So the interesting thing about this fibrous capsule that covers the testis is that it forms invaginations inside the testis itself. And these invaginations will divide the testis into partitions. It is said that each particular testis will have about 250, partitions, which are referred to as lobules. It is in these lobules, ladies and gentlemen, you're going to find tight coiled tubules where spermatogenesis will actually take place. These tightly coiled tubules are referred to as seminiferous tubules. I think we have someone trying to join now. Okay. All right. So these tightly coiled seminiferous tubules can be as long if they were connected, if you were to connect the seminiferous tubules in one particular testis, It can go as far as a kilometer, apparently, or roughly about 0.8 kilometers, almost about a kilometer. So we've already said that the spermatogenesis will take place in the seminiferous tubules, and once the sperms or the spermatozoa is produced. So I do have this diagram for illustration. So as you can see, this is the testis in blue, and you can see that inside this testis are partitions. You can see these partitions in blue, and you can see that you've got these yellow coiled tubes or tubules in these partitions or lobules, which is what should represent the seminiferous tubules. And we are saying that it is in this seminiferous tubules where you're going to have the production of spermatozoa. And once the spermatozoa is produced, it's going to be transported to the epididymis, as you can see. But in between the epididymis and the seminiferous tubules is a transitory passage, and which is referred to as the rete testis. Okay, so apparently in between, or shall we say, the partitions where you find these seminiferous tubules is a collection of blood vessels and interstitial cells. Okay, so in between these partitions. And these interstitial cells serve the role of producing androgens, sex hormones, which are referred to as androgens. And one of the important sex hormones in males is called testosterone, and it's very important in the process of spermatogenesis. In this diagram here, ladies and gentlemen, again, it's a section through the testis. So you can see that we're starting with, the scrotum, and you can see that we do have this layer of muscle, which we did say is the cremaster muscle, and just inside it we've got our scrotal cavity, and from the scrotal cavity we've got this fibrous covering of the testis, which is known as the tubica albuginea, and you can see that this tubica albuginea does traverse or invaginate the testis, forming partitions which are referred to as lobules, and we said there are about 250 lobules in each particular testis. And we did say that these lobules will have tightly coiled tubules where spermatogenesis is actually going to take place, and these are referred to as the seminiferous tubules. And we did say that once the spermatozoa are produced in the seminiferous tubules, they will be transported to the epididymis, but as a transitory point or as a passage to the epididymis, we have this network of tubules that are referred to as the rete testis. Also here, it's an actual image on a microscope of the seminiferous tubules at different stages of spermatogenesis. It's much easier to relate to this one. So this is the lumen of your seminiferous tubules, and you can see that here we have physically mature or morphologically mature spermatozoa, which is about to be released into the lumen of the seminiferous tubules. You can see that the other sections of this seminiferous tubules are showing us, sort of immature spermatozoa, basically. So this image is basically to illustrate the previous image if you couldn't see it properly. So this is like taking out one of the tubules, one of the seminiferous tubules, and you can see that the end product is spermatozoa, but the initial cells are referred to as spermatogonia stem cells, which are basically the germ cells, the germ stem cells of the seminiferous tubules, and are responsible for the production of the spermatozoa in the process of spermatogenesis. So it's very simple. So we can quickly talk about the spermatozoa itself, or the sperm. So it's basically made of three parts. It's got a head, middle piece, and the tail piece. The head is basically packed with chromosomes. It's got a cup, like one of those Nigerian cups. That is referred to as the acrosomal cup. The acrosomal cup is basically responsible for containing enzymes. These enzymes are important in breaking through, that jelly secretion on the ova during fertilization. The neck is basically packed with mitochondria, and we all know mitochondria is responsible for producing energy. And in this particular case, that energy is what drives the tail of the spermatozoa. The tail is basically made up of flagella, and it beats back and forth, and it helps to propel the spermatozoa itself, to put it very simple. So what we are looking at, ladies and gentlemen, without wasting a lot of your time, so this is the spermatozoa cell. You can see this is the acrosomal cap on top, and this is what we said contains the enzymes and is very helpful in breaking through the wall of the ova during the fertilization. You can see this black part is the head of the spermatozoa and will basically contain the genetic material contribution from the male. And you can see this middle piece is basically made up of mitochondria, which we said will produce the energy for driving this flagella, which is the tail of the spermatozoa. Okay, so it's important to mention that once this spermatozoa has been produced by the seminiferous tubules, when you look at it, it will look physically mature. However, at this point, it's incapable of carrying out fertilization. For it to be capable of fertilization, it must undergo a process of maturation, which is referred to as capacitation. Okay, as you're going to see in the slides. Yeah. So before we talk about capacitation, we can briefly talk about the organs of the male reproductive system that will help us transport the spermatozoa all the way to the external meatus of the urethra. Okay, so the first one is the epididymis. So like we already said, so after production in the seminiferous tubules, the spermatozoa, it will be transported to the epididymis through the rete testis. The epididymis is basically made up of elongated tubules, roughly about 7 meters. It is tightly packed in such a way, if you can see from the drawings, you can imagine 7 meters, fitting in that small space. So, it's really, really tightly packed. It takes about 2 weeks for the spermatozoans, which is immature spermatozoa, to travel through the epididymis. As they do, they will be undergoing maturation. But also another important purpose of the epididymis is the fact that the epididymis will remove um, or destroy any damaged or abnormal spermatozoa, and the components of which will be recycled for making new spermatozoa in the seminiferous tubules. So we did mention that the spermatozoa from the seminiferous tubules are basically in a still state. So they're physically, they look mature, but their flagella is not beating. The epididymis does play a role in the maturation, but even at this point, they're still inactive. And we did say that for them to become active, they must go through a process which is known as capacitation, and it will happen once they interact with the secretions from the seminal vesicles, but also after exposure in the vagina environment. Okay, so we've already said that. So from the ductus deferens, or rather from the epididymis, the spermatozoa will be transported to the ductus deferens. Now, the ductus deferens is basically a tubular structure. It is said to be about 5 meters long, so it extends between the epididymis and the urethra, or if you like more specifically, between the ejaculatory duct, like we already said at the beginning of this talk. So it doesn't travel alone, it's like Liverpool, doesn't walk alone. So it basically will be found in a sheath of connective tissue, and in this sheath of connective tissue will be other structures. These other structures include blood vessels, nerves and lymphatics, and clemaster muscle. Now, the blood vessels, which is obviously artery and vein, and nerve supply the testis. All right. Now, this whole collection of spermatic cord, artery, vein, nerve, and lymphatic form what is known as spermatic cord. In my days in medical school, this was a common question. They would say, tell us the 10 components of the spermatic cord. But I don't think that is a valid question for this course. Anyways, so what we're looking at, ladies and gentlemen, is basically a drawing. So we are looking at this, you can say, if we're to visualize a camera, so it's like the camera is viewing the back of a patient. It's back, so it's like as though, it's what is the best to describe? So the position is you're looking at somebody from the bum, and you have the bums above you, like that. So you can see that what we are looking at posteriorly, this is the back of the urinary bladder, and inferior to the urinary bladder we've got the prostate, and you can see that we've got two ductus deferens or spermatic cord, if you like. No, ductus deferens is better, not spermatic cord. So ductus deferens that is coming. So as it's coming, it's joining the duct that is coming from the seminal vesicle, and when it joins the duct that is coming from the seminal vesicle, it forms what is known as the ejaculatory duct. The ejaculatory duct is a very small, about 2.5 centimeters, and it pours into the prostatic urethra, as you can see. So it's very easy. Okay, so I think we've already said this. So the ductus deferens itself is made up of a muscular wall, which is smooth muscular wall, and like all other structures that are made up tubular structures that are made up of smooth muscle, it undergoes peristalsis. I'm sure you are familiar with peristalsis from the GIT and from the ureters. Oh, we haven't discussed the urinary system. We will very soon. So it is these peristaltic contractions in the ductus deferens that will help to propel your spermatozoa from the epididymis side all the way to the urethra, basically. Not only is it responsible for the transportation of the spermatozoa between the epididymis and the urethra, but it also can serve the purpose of storage of the spermatozoa, and this can happen over several months. Okay, so like the epididymis, this spermatozoa at this stage in the ductus deferens is immature and has not undergone the process of maturation yet. I think we've already discussed the ejaculatory ducts, so I shall not waste time there. So from the ductus deferens, we move to the urethra. We've already shown how the ductus deferens joins the urethra, and we said that it will join the urethra as an ejaculatory duct. The ejaculatory duct is basically a small passage of the ductus deferens that forms as it is joined by the duct from the seminal vesicle, and it is about 1 inch or 2.5 centimeters. So the urethra in the male is quite long, so it extends between the urinary bladder to the tip of the penis. It measures on average between 15 to 20 centimeters, and it serves as a passageway for semen, but also for urine. Now, you may wish to know that the secretion from the testis forms a very small portion of the ejaculate. The ejaculate is basically, the volume, or shall we say, the material that is released, after every sexual, excitation, at the climax of sexual excitation, sometimes also referred to as semen. So what is produced from the testis only forms about 5% of the final volume. So where does the rest of the volume come from? This is what we are going to discuss very quickly. So it comes from what are known as the accessory glands. And the accessory glands, like we've already mentioned, are made up of the seminal vesicles, the prostate gland, the bulbourethral gland, also known as the Cowper gland. Let us dwell into the details of this. So what is the purpose of the secretions from these glands, the accessory glands as we've called them? So basically is to activate the spermatozoa. Remember that up to this point, from the seminiferous tubules, the epididymis, the ductus deferens, these spermatozoa are inactive and their flagella is not beating and they are incapable of fertilization. Right. So one of the important functions is to activate or mature the spermatozoa in readiness for fertilization. It is also to provide nutrients for the spermatozoa. Um, um, it also assists in propelling the spermatozoa along the reproductive tract through peristaltic contraction. They also provide buffers. As you know, the vagina in particular, is acidic, and so to counter the acidic secretions of the vagina for the spermatozoa to survive, most of these accessory glands, their fluids are alkaline, except the prostatic gland, as you are going to see later on. So we'll start with the seminal vesicles. You may remember the seminal vesicles. So we are basically talking about these two glands. There are two glands just at the back, posterior and inferior surface of the urinary bladder. So these seminal vesicles are said to be about 5 centimeters in length. They are said to contribute about 60% of semen or ejaculate. This ejaculate will be made up of fructose. Fructose is basically the sugar that the spermatozoa can easily metabolize to produce energy. And like we've already mentioned, this secretion from the seminal vesicles is alkaline. And the reason it is alkaline, it helps to neutralize, the acidic vagina environment, but also the prostatic secretion, which is also said to be acidic. Once the spermatozoa comes in contact with the secretion from the seminal vesicles, it now becomes active and starts to breed its flagella and becomes highly mobile. You could say that maybe between that time, of course it's a much complex process, but as a way to remember it, you may think that, well, maybe before the spermatozoa meets the secretion from the seminal vesicles, you could say that it was starving. It didn't have the fructose, didn't have the food. But once it gets the food, it now becomes very active and ready for action. I think we've already looked at this structure basically, so we know the seminal vesicles. We already talked about the ductus deferens. We already talked about the ejaculatory ducts. We already talked about the prostate gland, and we already talked about the urethra, in particular the prostatic urethra. So there's nothing new here. So we can quickly speak about the prostate gland. So the prostate gland is basically a small muscular rounded organ, which we've already seen. It is said to be about 4 centimeters in diameter. It surrounds the urethra just as it comes out of the urinary bladder, as we can see in this diagram. And we know that this part of the urethra is referred to as the prostatic urethra. Okay. So just like the seminal vesicles, the prostatic gland also does secrete, secretions which contributes to the semen, and it is said to be about 30% of the final ejaculate in volume. And we did say that it is acidic, but the secretion is important in the fact that it contains seminal plasma proteins or seminal plasmin. These seminal plasmin are important in the fact that they possess antibacterial properties. Now, these antibacterial properties are important in males to prevent urinary tract infections. apparently. Um, so what will happen is that the secretions of the prostate gland um, will be propelled to the urethra um, by contraction of the muscular wall of the prostate gland. So another important gland which contributes to the final volume of the ejaculate is what is known as the bulbourethral gland or Cowper's gland. So this one, like the seminal vesicle, produce an alkaline mucus fluid. And obviously, we did already say that this one is important in helping to neutralize, the acid in the vagina, but also the acidic secretion from the prostate. Additionally, the Cowper's gland secretion is mucus, and because it is a mucus in nature, it does, help in lubrication. In fact, it is said that when a man is aroused, there's that initial secretion that you get from the penis. That secretion is not semen, but rather from the Cowper's gland. Okay, basically. All right. So we can quickly talk about the ejaculate or the semen itself. So the ejaculate is said to be around, about two to 5 mils. So, and this is the fluid that is basically released at the height of sexual arousal. So it contains about 20 to 100 million spermatozoa per male. So if it is 2 males, we are saying at most you should have 40 million, spermatozoa. And if you're producing five, so you should have a bare minimum of at least 100 million or 500 million, at maximum. Is there a question, or are we running out of time? We are running out of time. I'm almost concluding. Should minimize the too much talking. All right. Okay. So, my thoughts on this are that, well, colleagues, if you're feeling bad about yourself, you must always remember that you are one of the 500 million or the 200 million that succeeded, in because you can imagine you are one of the few, you're the only one, as a matter of fact, in that particular ejaculate of the maybe 200 million. 200 million is like the population of America or Nigeria that succeeded to be alive. So if you ever feel bad, at least that should console you. So anyway, apart from the spermatozoa, semen is also made up of the seminal fluid, which we did say is a mixture of the various granular secretions with a distinct ionic and nutrient composition. In addition, there is also enzymes, which are basically proteases, and they help to dissolve the mucus secretion from the vagina. So we can also talk about the external genitalia. We've already started when we talked about the scrotum. We can now just conclude by talking about the penis. So the penis is basically the tubular organ that's around the urethra. It conducts urine to the external environment, and also introduces semen into the vagina during sexual intercourse. So it is made up of the root, made up of three parts: the root, the body, and the glands. The root is basically the part that is fixed and attached to the body. The, the body is a tubular portion that is made up of erectile tissue. We don't have the glands here. No. But we should have the glands. Anyway, it is made up of three parts. The glands should have been here. Oh, it is here, as a matter of fact. So we have the glands there. So the glans is the expanded distal end, surrounds the external urethra or the external urethral meatus. At the glans are folds of skin that surround the penis, and this is referred to as the prepuce. Yes, the prepuce. It is this prepuce that is cut during circumcision. Now, the prepuce does secrete as glands, which have a waxy secretion. This waxy secretion is known as smegma. Now, smegma, unfortunately, can be very good nutrients for bacteria. And unfortunately, if you do not clean your, at this age, I mean, we're probably big people. My audience is big people, so you know about this smegma. So we know that you must clean it, because if you don't, bacteria will feed on it, and then you might get infections and things like that. And of course, if you are incapable of regular cleaning, or you do have some anatomical abnormality which maybe prevents you from getting rid of this smegma, perhaps a solution would be circumcision, where you remove this foreskin for good, so you don't have to deal with those secretions. Basically, this is a drawing of the penis, the different parts. This is the glans, this is the body, this is the root. And you can see that this particular man looked as though he's been circumcised, because we cannot see his prepuce or foreskin. And you can see that we have this corpora cavernosa, which is part of the erectile tissue of the penis. Right, so speaking about the erectile tissue, so this will be found in the shaft or the body of the penis, and there are three columns. So on the anterior surface of the penis, you have two cylindrical, as you can see, on the anterior surface, you've got this two cylindrical erectile tissue, and is referred to as the corpora cavernosa. So the corpora cavernosa is bound to the pubis and the ischium of the pelvis, and it extends all the way from the pubis and the ischium to the glans of the penis. So apart from the corpora cavernosa, we also have the relatively slender corpus spongosum. The corpus spongosum is you could say, is on the posterior surface of the penis, and it is the erectile tissue which surrounds the urethra. If you look at this diagram, which is a section through the penis, you can see that here we've got so this is so it's more like we are looking at the penis from, imagine the camera is on the ground, and we are looking at the penis as though, with our eyes looking at the sky, and the penis is outstretched. Oh, I didn't show you the position of the bulbourethral glands or the Cowper's gland. So you can see that this is our a corpus spongosum, and here we've got our glands, sorry, what am I saying? We've got our corpora in blue, corpora cavernosa, which are the two cylindrical erectile tissue. And we've got this singular erectile tissue, which is known as the corpus spongiosum and surrounds the urethra. You would appreciate it better here if, God forbid, your penis was cut. This is how it would appear. So you can see that we've got our corpus spongiosum and we've got the two corpora cavernosa. You can see that the corpus spongiosum does surround the urethra, and you can see that we do have a central artery in the corpora cavernosa. So something that we already know. So we know that, well, in the resting state, these blood vessels, or the blood vessels that supply blood to the erectile tissue, the corpora cavernosa, and the corpus spongiosum, will be, how can you say, the blood flow will be restricted, or the walls will be narrowed. But during sexual arousal, you will have activation of the parasympathetics, and not the sympathetics, but the parasympathetics. It is these parasympathetics that will lead to the dilatation of the arterial blood vessels that supply blood to the erectile tissue. And obviously, because it is erectile, it's like a balloon. If you inflate it, it becomes bigger, and that is what leads to an erection during sexual arousal. Of course, we know, as you grow older and you get this atherosclerosis in your blood vessels, or maybe you are diabetic, your blood flow is not so good, or your valves cannot, return the blood in your erectile tissue. This impact would lead to, or could explain, erectile dysfunction in patients such as this. Good thing is that in radiology we can actually determine blood flow. Let's say in patients that have erectile dysfunction, we can determine blood flow, in these blood vessels, and recommend appropriate treatment. With that, ladies and gentlemen, I end the first part of this talk. I don't know if you have questions for me. There are no questions?
Nope.
Okay, if there are no questions, I should be able to finish the female reproductive system within the course, I think maybe tomorrow or Tuesday. And then we'll quickly cover the urinary system and the nervous system, and I think we'll be done for the semester. And I think we should have some time to revise. I'll bring some questions that we can discuss, and then I will share with you what I think are appropriate answers. We see what we can learn from each other. Okay, guys, enjoy.
Thank you.
Okay, bye.
Thank you.